So indeed, what I will tell you about is the role, the coupling between energy metabolism and synaptic activity. In fact, this has been one of the fundamental principles first proposed in the end of the 19th century by Charles Sherrington, and I will just mention this in a moment, to show that there is indeed a coupling between the activity that neurons display and the delivery of energy to the active neurons. And this has been, by the way, the basis for functional brain imaging techniques, because what is monitored is actually the consumption of energy related to neuronal activity. Actually, Sherrington already proposed for vascular coupling, meaning bringing more substrates to the active area, that the brain possesses an intrinsic mechanism by which its vascular supply can be varied locally in correspondence of local variation of functional activity. And this is over 130 years ago, very forward-looking, but no idea of the mechanism, just a description, but very important one. And then in the 60s and 70s, Lou Sokolov showed that not only this extra energy that comes when the neuropil is active actually is also the case for metabolic coupling. In other words, glucose utilization. And this is a very famous image showing ocular dominance column in the primary visual cortex showing activity related and consumption of energy. So in summary, the basic principle here is whenever a group of neurons is more active, it will be using more energy. And the question was, what are the mechanisms for this coupling? And my group, and later many others, showed that actually a key cell type in the brain, the astrocytes, which belongs to the general category of glial cells, plays a key role in this coupling. Everybody knows neurons. They're the noble cells of the brain, but half of the brain in the human are actually glial cells, and most of them astrocytes. And these astrocytes have particular features that make them very fit to couple neuronal activity to energy delivery. For example, this is an old drawing by Santiago Ramón y Cajal. You can see that this astrocyte here has some process that is very close, at the time one couldn't tell how much, but very close to the capillaries. And then you see also that these astrocytes, although they have rather limited processes, they actually are very packed. So they can actually cover a given volume of cortex or any other area of the brain. These are more modern pictures. This is actually a synapse, a 3D reconstruction of a synapse, the contact between neurons. And this is the presynaptic profile, the postsynaptic, and here are in yellow, in blue, some processes from astrocytes that somehow ensheath the synapse. This is a dynamic ensheathing, but it shows how close they are to the action. So the astrocyte can sense when synapses are active. And also, this is another picture from my lab. You can see here an astrocytic process called end-foot that is actually on the capillary. So with a more artistic view, you can actually visualize here an astrocyte that essentially covers the capillaries, the source of energy, with this end feet, and also covers or somehow ensheath synapses, the site of where neurons exchange information. And not only are they in the right place, but they also express the appropriate molecules, because at the synapse, astrocytes express receptors and reuptake sites for neurotransmitters, in particular glutamate. This is great work by Jon and his group over the years. And here they express transporters for glucose, of course, but also for amino acids and monocarboxylates. So the question that I asked was, the question that my lab has asked was, again, what are the mechanisms that allow this coupling? And what I will review, and as I said, the key cells here are astrocytes, these glial cells. And I will actually review 4 aspects in my lecture. First, the implications for neuroenergetics, in other words, brain energy metabolism, for functional brain imaging, for neuronal plasticity, and eventually on disease. So the first evidence for this coupling came during my PhD. You can see me when I was 27 or 28. Things were different at the time. You could go in the lab with shorts, no white coat. Well, anyway, many things have changed, as you can see. And this is work that I did with my mentor, Floyd Bloom, a great man who unfortunately is not in good shape now, but he was really an inspiring person. And there we showed that VIP, a peptide that was very recently discovered in the brain, we are talking about the late 70s, early 80s, was promoting a kind of an endocrine type of effect, promoting glycogenolysis. Glycogenolysis immediately points to astrocytes. This is an EM picture of the brain, where you see the capillaries here. And then you see this process here with electron-dense granules that are glycogen. And glycogen, which is the storage form of glucose, very rich in liver, also in muscle, in the brain, not much, but whatever glycogen is in the brain is here in astrocytes. And this is the neuropil with synapses. So we showed that actually signals from neurotransmitter from neurons, meaning VIP, we also showed noradrenaline and later adenosine, can break down glycogen, provide energy to neurons. So that was the first formulation in 1981 of the existence of a coupling between neuronal activity mediated by VIP or noradrenaline and energy metabolism that provide energy to neurons. And by the way, at the time, and this is a picture, again, of those years. With John Morrison, still is a very fine neuroanatomist, we proposed that actually the VIP promoted glycogenolysis was restricted to cortical columns because VIP neurons are these very radial bipolar cells that act very locally. And then that was to be contrasted with the noradrenergic system, which, as you know, originates in the locus ceruleus, and from there, fibers project throughout the brain. So the idea was that whenever there is an activity, modality-specific activation, VIP neurons will be activated, and when there is an event that primes metabolically the whole cortex, like when noradrenaline is active, for example, during attention, then this would become operative. A few years later, in Lausanne, I was back in Lausanne with Luc Pellerin, who was just joined my lab from Montreal. We showed that actually the main neurotransmitter in the brain, glutamate, was also having a metabolic effect on astrocytes, namely aerobic glycolysis. So what is aerobic glycolysis? Is the utilization of glucose and production of lactate in the presence of oxygen. This is a well-known metabolic process known as the Warburg effect. And lactate, as Linda alluded to, was long considered bad, garbage, but actually, because it's associated with hypoxia or ischemia. But in the presence of oxygen, as I will show you, it is a very important energy substrate and signal for neurons. So we proposed this mechanism to couple glutamate-mediated synaptic activity. So you have here a presynaptic vesicle releasing glutamate, which will act on postsynaptic receptors and excite neurons. But as I mentioned earlier, in particular, the group here in Oslo showed the importance of this reuptake of glutamate from the astrocyte, one of the main functions known at the time for astrocytes. And this glutamate has taking up glutamate has two consequences. First, it's converted, part of it at least maybe 70, 80% to glutamine, and this uses one ATP. And this glutamine then, and again it's been studied here in Oslo, it's taken up by, recycled in synaptic terminals and then converted back to glutamate in order to replenish the glutamatergic pool. And then the other mechanism that is associated with glutamate uptake is an increase in intracellular sodium in the astrocyte, because the transport of glutamate into the astrocyte is driven by the electrochemical gradient of sodium with a rather precise stoichiometry of 1 glutamate with 3 sodium ions. So essentially, sodium increases in the astrocyte, and again, like in any cell, the homeostasis of sodium is reestablished by the activity of the sodium-potassium ATPase, in particular the alpha-2 subunit. And so essentially, whenever astrocytes take up glutamate, they will have to spend energy. And this will activate, like in any process in cells that are exerting activity, it allows the uptake of glucose through different mechanisms, and essentially transporter and phosphorylation. And then was the big surprise for us, lactate is essentially produced from this glucose. There is still, of course, uptake of glucose into neurons, but during activation, it's mainly through this process. And by the way, there are specificity here. The glucose transporter in astrocytes and capillary is GLUT1, and the one in neurons is GLUT3. So these are different entities. So this gave rise to what is now known as the Astrocyte Neuron Lactate Shuttle, whereby a neuronal signal, and as you know, glutamate is essentially released at 80% of the synapses in the brain. So whenever there is activation of any modality, there will be glutamate released. It is taken up by astrocyte. This triggers the mechanism I described with glucose uptake. Then this glucose is converted to lactate mainly, and then transported through specific transporters from astrocytes to neurons. And then in neurons, in the presence of oxygen, it is converted to pyruvate, and then it can provide energy for the respiratory chain mitochondrial activity. So essentially, neurons are mainly oxidative, and astrocytes are mainly glycolytic. Since the initial discovery, actually several other candidates have been identified for this type of coupling, mostly by the lab of Felipe Barros. For example, potassium, which is also increased by neuronal activity, ammonium can trigger this shuttle, and then actually even it seems to exist also a channel with high capacity for releasing lactate. Now, studies that came later show that indeed, if you look at the genes expressed in astrocytes and neurons, they really, and it's not surprising, are exactly subserving this glycolytic activity in astrocytes and oxidative activity in neurons. Essentially, astrocytes express this enzyme, phosphofructokinase fructose bisphosphatase 3. You have to be a biochemist to be able to pronounce it. In neurons, it is present, but it's immediately degraded by proteasome activity. And then astrocytes express this pyruvate kinase, the M2 form, which is present in tissues that are glycolytic. And the activity of pyruvate dehydrogenase, the enzyme that actually uses pyruvate, is actually low in the astrocyte. In neurons, essentially, as I said, there is no activity of PFKFB3. As I mentioned before, there is no glycogen. And the activity here is PKM1, which is typical of tissues that are oxidative. And PDH activity is quite high. So there is, obviously, it's not surprising, a level of expression of the enzymes and proteins that matches the functional activity. So this is really for the first component of neuroenergetic. The message is there is a metabolic coupling between neurons and astrocytes whenever neurons, glutamatergic, but also modulatory neurons, such as VIP-ergic and noradrenergic, are active. They act on astrocytes to provide energy to neurons. Now, how is this relevant to functional brain imaging? Well, we actually addressed this in experiments about 20 years ago. We said, well, if this reuptake of glutamate into astrocytes is so key, crucial for glucose uptake, if we were to downregulate the expression of these transmitters, maybe we will decrease the uptake of glucose into the brain. So we did experiments in vivo in this well-known neuroanatomical physiological pathway, the whisker to barrel. So for each whisker, there is a corresponding aggregation in layer 4 of somatosensory cortex called the barrel. And you can actually selectively stimulate a vibrissa and then see an increase in imaging of glucose with 2-deoxyglucose autoradiography in the corresponding barrel. So it's a nice system to study coupling between activity and glucose utilization. Well, when we actually downregulated the expression of astrocytic glutamate transporters, what you see, you can focus on this last panel. This is the control. You see a nice increase in glucose utilization. But in the corresponding barrel where we downregulated the expression of glutamate transporter, you essentially see no image of glucose utilization, meaning that this mechanism is a key for glucose uptake into the brain. And other studies, for example, by Yuri Bujaki, showed that this is the visualization of glucose uptake during activation, in vivo also. Before activation, you essentially, and you look at fluorescent glucose in the various compartments. This is blood, this is astrocyte, these are neurons. And during activation, you actually see the increase essentially in astrocytes, not in neurons. So there are many other evidence. I will not spend too much time on this, but essentially the message here is that when using the fluorodeoxyglucose positron emission technique to visualize activity in a given brain area or throughout the brain, the signal essentially during activation, maybe also to a certain degree also during basal activity, actually originates from the astrocytes. It doesn't detract anything to the technique because the original signal is a neuronal one. So it's fine to localize, but one should keep in mind that one sees a FDG PET image, actually most of the signal is coming from the astrocyte. And some time ago, a group at McGill showed that doing the opposite, in other words, increasing the expression of the glutamate transporter resulted in an increased PET signal in rodents. So now let me move to the more recent work on neuronal plasticity first and then on disease. So a few years ago, we asked the question. Actually, it was just before I came here 10 years ago to give a lecture at the Academy here. We said, well, now that we have described this, let's call it cycle, the shuttle, this coupling, what happens during learning, during neuronal synaptic plasticity? A lot is known about synaptic plasticity, an increase in the postsynaptic receptors, an increased release, gene expression. But all these observations are neuronal. So we said, does this coupling adapt during plasticity? Or maybe is it even necessary for plasticity? And so we undertook a series of time-consuming, very time-consuming experiments to try to actually look whether there was a component in plasticity or an adaptation and a component in plasticity related to the astrocyte neuron lactate shuttle or to its neurometabolic coupling. So what we did, we subjected rodents to different behavioral tests, either spatial memory or what is called inhibitory avoidance. I'll tell you a bit more in a moment. Then mapped the 2-deoxyglucose before and after learning. So that gave us an indication of which areas are mobilized during this type of learning. And then we laser microdissected these areas and looked at expression of genes in this microdissected area where glucose consumption is increased after learning. And this is work by Monica Tadi, and we show that indeed a number of genes are upregulated in the areas that were mobilized during learning. And interestingly enough, of course, there is phosphofructokinase, pyruvate carboxylase, but essentially all the enzymes related to glycogen metabolism. This was in the maybe 2008, 9, 10. So it brought me back 20, 25 years when I showed you when I was in shorts in the lab working on glycogen. And so the idea was, well, if glycogen metabolism is so important for learning, how about if we block glycogenolysis during learning? This is a purely astrocytic mechanism, and see what happens in learning. So is there plasticity or not? And so, and also we blocked the transport with oligonucleotide antisense. We decreased the expression of these monocarboxylate transporters. 1 and 4 are astrocyte specific, 2 is neuronal specific. And this is work I did with Cristina Alberini. She's really an expert in the molecular mechanisms of learning. And we published this now 12 years ago, and essentially showing that astrocyte neuron lactate transport is required for long-term memory formation. This is a pretty counterintuitive statement because, you know, neurons are supposed to be the noble cells of the brain, and lactate and astrocytes, yeah, maybe, you know, a friend of mine, Dean of the Collège de France, told me one day, Pierre, you have become very visible and made contribution by studying the garbage of the brain, lactate and glial cells. So anyway, what did we do? We injected bilaterally in the hippocampus this molecule, DAB, that blocks glycogenolysis, essentially glycogen phosphorylase. And then, so we blocked here. And then later also we blocked, decreased the expression here and here. And then tested long-term memory in this apparatus or behavioral paradigm called inhibitory avoidance. In other words, you place the mouse in a two-compartment cage. One is lit, one is dark. And as you know, rodents like dark, so they quickly move to the dark compartment. But the day of training, you give them a little electric shock when they move into the dark compartment. So the next day you test them. And if they remember, they will take a long time before moving into the dark compartment because they remember something unpleasant happens there. And so a long latency means good memory. A short latency means that memory has not been formed. And you see here the control situation. You see, again, the latency is long. And if you give vehicle, nothing really happens. But if you look at here, if you co-inject the inhibitor of glycogen phosphorylase, so you prevent the formation of lactate, and you exogenously administer lactate, well, you rescue memory. So essentially, it happens the same when you downregulate the expression of MCT1 or 4, so preventing lactate to move from astrocytes to neurons, you block memory, and this is rescued by lactate. When you downregulate MCT2, the neuronal one, you also block memory consolidation, but it's not rescued by lactate, which is not surprising because lactate cannot enter properly in the neuron. And this is just to show you that there is indeed an increase in extracellular lactate during learning that disappears when you block glycogenolysis, which happens in astrocyte. This is a bit specialized. It's just to show you that long-term potentiation, which is a cellular form of memory, well established, actually initially discovered also here by Andersen. And Lømo, yeah, exactly. Here in Norway. It means that you can facilitate a synapse long-lasting, but if you actually block glycogen mobilization and lactate production, you can still induce LTP, but you cannot sustain it, meaning that in cellular model you cannot. And this is in vivo work. So in summary, what this experiment showed that glycogenolysis and astrocyte neuron lactate shuttling are required for long-term memory formation. An interesting and surprising observation, which we followed up, and I will show you in a few next slides. We also wanted to check whether this was only true for aversive memories, pain, like the inhibitory avoidance, or also present in appetitive memory formation in another area of the brain, such as the amygdala. And so we looked at what is called conditioned place preference for cocaine. So essentially, you have also two compartments, very different in terms of texture of whatever is present in that compartment. One, you give cocaine a few times, then the other you just give sugar. And then a few days later, you test the preference, and then the rodent goes where it got cocaine. So it's called place preference. And this is work by this group. Actually, I wasn't there the day of the picture, so they were kind enough. They put me in a picture in the background. But anyway, this is the place preference. So it means that if the experiments classically works, you will see the mouse moving into the compartment where it received cocaine. If you actually give DAB during learning, no preference. And then you give lactate, back preference. You can, they again show preference, but pyruvate doesn't do it. So another way of showing that memory, that plasticity is linked to this transfer of lactate from astrocytes to neurons. Then we ask the next question. If glycogen is so important in memory consolidation through glycogenolysis and the production of lactate, where is glycogen localized in the astrocyte? So this is, again, very time-consuming work by Corrado Calì, who's now a professor at the University of Torino, and many of master students and interns that came over 3, 4, 5 years even. It really was a very long study, where he did serial EM sections and then 3D reconstruction, and did what is known as segmentation. So on each section, you identify what is a profile corresponding to, either an axon, a dendrite, a capillary, that is easy, or an astrocyte profile. And so this is actually something that was done at KAUST with a really amazing facility called the Cave, where you can, and with the help of a computer visualization group, where you can transform all this information into a 3D virtual reality environment. So in a way, you can take a stroll into this volume of cortex. And that's what that helped us to understand better the relationships. So here it's simply yeah, the elements. You can see in green, by the way, the astrocytes, to see how they are pervasive in terms of processes, the axons, the dendrites. And these are glycogen granules that you can identify in each astrocytic profile. And this is Corrado in the Cave. And he's actually literally, or virtually actually, inside a capillary here. And then you see these are data. You see down there an EM section. And then he moves out of the capillary and now moves into the astrocyte. He saw, he's moving. Okay, there we go. And then you can, you see all these glycogen granules. You can render transparent the astrocyte, and you see spines of neurons outside the astrocyte. And so, of course, it's kind of cute and nice, but it really was very useful because it showed, in a way that could not be seen in 2 dimensions, frankly, or possibly, but much more difficult, clusters of glycogen. And then when you analyze quantitatively, you see that essentially 2/3 of the glycogen granules are in astrocytes facing varicosities or dendritic spines, so where the signal for glycogenolysis will come. And following up on this, we looked, Corrado and some colleagues in Milano, looked at a particular type of learning, novel object recognition. And you see here the number of spines per cubic millimeters, and something that was known actually, but there are not, if you look closely, there are not that many papers. There is an increase in the number and also in the volume of dendritic spines after learning. But for us, more interestingly, you have an increase in glycogen granules, in number and size of glycogen granules facing these expanded dendritic spines. So it shows that there is a signal that goes to astrocytes, and there is more glycogen available. We don't know yet exactly the significance of this, but in summary, this is a basal condition. You have spine and then some glycogen close to in front of the spine. After learning, you have an increase in number and volume of spines and an increase in number and size of glycogen granules in the astrocytes. If you block learning with DAB, like we showed before, you don't have the increase in spines and you don't have the increase in glycogen granules. And if you give lactate back, you actually have again an increase in size and particularly number of the spines, not so much the size. Not on glycogen granules, but that's not surprising because glycogen doesn't form actually glycogen. So the summary is that, yes, there are the key element, glycogen, in astrocytes is actually necessary or at least associated with learning and an increase in spine size and number, which is known to be associated with learning. So this, we're now trying to understand the signal here. Now, the question then was, okay, well, lactate, of course, you remove lactate production, so maybe you don't have fuel. So no surprise that you don't have memory consolidation. So what we did was to replace, after DAB injection, lactate I show you, but glucose. Glucose doesn't do the job. It doesn't do the job like lactate. And in fact, if you look at increase in gene expression, for example, that are related to learning, and this is work done in vitro, you can see that lactate, but not pyruvate, not D-lactate, not D-glucose, increase the expression of these genes, ARC and ZIF in particular, and BDNF, not the other energy substrates. So there is something special about lactate. And so we looked at trying to understand the mechanism through which lactate was specifically involved in learning and memory consolidation. And eventually we found that actually lactate promotes the expression of a number of genes involved in plasticity by actually increasing the activity of the NMDA receptor, which in a way is not so surprising because NMDA receptor is key for memory consolidation. And work that we did further showed that actually lactate, again, so lactate enters through MCT2, it's converted to pyruvate, it produces energy, but also through NAD and ADH, NADH modulates the activity of the glutamate NMDA receptor, and then one of the consequences is a regulation of some expression of genes. And so what we saw is that actually a number of genes are upregulated, genes involved in synaptic plasticity and neuroprotections, and a few genes are downregulated, and these are genes associated to cell death. So that will bring me to the final part of my talk related to the role of lactate and neuron-glia metabolic coupling in neuroprotection in addition to memory. And this is very recent data. What we see is that actually lactate increases the peak of an NMDA current. And so therefore it means that simply the NMDA current is much larger, and that triggers, among other things, the induction of genes. So in summary, this work over the last 10 years, let's say, shows that lactate is not only an energy substrate in the presence of oxygen, but it is also a signal for plasticity in particular. And very recently, we showed that, in fact, when there is a low-level stimulation in plasticity, again, it's a bit technical, spike time-dependent plasticity, glucose is sufficient. But when we go higher, in particular theta burst stimulation, this is really the stimulation that is involved in memory consolidation, lactate is necessary. And similarly for behavioral tests. So let me get to the final part of my talk about diseases, how this neuron-glia metabolic coupling is involved in diseases and potentially opens for therapeutic interventions. And in fact, I will show you some examples whereby glucose uptake and lactate production have an impact on potentially on mood disorders, specific rare genetic disorders, GLUT1 deficiency syndrome, and also potentially on neurodegeneration and Alzheimer's. So the reason why we did these experiments that I'm showing here is we showed that actually peripheral administration of lactate produces antidepressant-like effects. And the reason is that there was evidence that glial signaling is involved in depression models, also in a decrease in brain banks for patients died who suffered from major depression. And also, as I showed you, there is an increase in expression of a number of genes. And depression, in a way, is a form of disease of plasticity. And so we saw, won't go in detail, but we used 3 models, acute forced swim test, open space forced swim test, and chronic corticosterone, 3 models of quote-unquote depression in rodents. And we could counteract the behavioral effects, but also, by the way, a number of molecular changes were normalized after lactate administration. And more recently, we showed that, in fact, for this effect of lactate to take place, there is need for neurogenesis to be active. And this is just to show you that this is controlled. This is after chronic corticosterone. These are a number of these neuroprogenitor cells that are involved, of course, for neurogenesis. And then lactate counteracts this negative effect on neuroprecursor cells. So let me go very briefly to 2 other diseases, GLUT1 deficiency syndrome. And by the way, the data that I will be showing here are from a spin-off from my lab at EPFL called Glia Farm. So I have a conflict of interest. This disease, glucose transporter 1 deficiency syndrome, as it says, it's a mutation of the glucose transporter, which is selectively present in astrocytes and capillaries. And as you can see, it results in epilepsy in the young age and then important motor dysfunctions. And you can see a decrease in the PET signal in these kids, and interestingly, a decrease both of glucose and lactate in the cerebrospinal fluid of these kids. And so essentially, this astrocyte neuron lactate shuttle is somehow downregulated. And the idea is with molecules that were developed through a number of phenotypic screening by this spin-off company was to boost the astrocyte neuron lactate shuttle, to boost glucose uptake and lactate production in vivo. And this is work those who collaborated, including Daryl DeVivo from Colombia, who actually is the one who described this disease. And essentially, there is a decrease. You look at all these bars here. There is a major decrease in glucose, lactate, and glycogen in the brain of these mice that have this disease, transgenic mice, of course. And this molecule that was developed by Glia Farm increases glucose uptake after oral administration, measured by biosensors, and also lactate. It also normalizes the PET signal in these mouse mutants, and it improves significantly the motor dysfunction that these mice suffer of. So the concept here is really to provide more glucose, provide more lactate, in other words, to provide better fitness for the brain in conditions where there is a decrease in glucose utilization and glucose availability. The idea being that you decrease the threshold if you have an energy crisis, you decrease the threshold for any insults to be neurodegenerative. And this is just very recent data on aging mice. This is a classic behavioral test, the Morris water maze. Young mice go very quickly to the platform where it is hidden. And the old mice, they take a long time because they don't learn. And so the distance, long distance meaning you don't learn, short distance you learn. This is in aged mice. And then this is in a model of Alzheimer's, the streptozotocin, also again, short distance, it means they do much better. So in conclusion, this work really has addressed many facets of neuron-glia metabolic coupling, showing that there are a variety, but very few in a way, neurotransmitters, noradrenaline, VIP adenosine, so modulators of glutamate, the main neurotransmitter in the brain, that eventually lead to lactate production by the astrocyte, and this has effects on neural plasticity, memory, neuroprotection, and mood. And this is actually work that's really started a bit by chance when I was in shorts in the lab, working on glycogen. That brought me to become interested into astrocytes, to lactate, plasticity, and now memory, and possibly this cycle, the Astrocyte Neuron Lactate Shuttle, being a therapeutic target. It took about 40 years, and I can look back and I'm quite, rewarded by this, and look forward, actually, to do more. This is of course, this didn't happen by myself. There are many, many, many collaborators at EPFL, at the University of Lausanne, in KAUST, many collaborators. And I look forward again to more of this. And then these 40 years, about 30 of them, not continuously, also inspired a book that I wrote with my wife, Christine, Ammons Horn, which is about Alzheimer's and neuroscience, which is coming out in a couple of weeks. And so you see, science can bring you to many destinies. Thank you for your attention.
We have planned for a panel discussion and would like Lene Juel Rasmussen, who is the director of the Center for Research on Healthy Aging in Copenhagen, who has given a Nansen lecture before, but that was during the COVID, so she couldn't be here. And now she's here, so she can join into the discussion. And I would like to start by asking Lene to open the discussion.
When I gave the lecture here, the title of my talk was Understand and Intervene. I'm a molecular biologist, so energy metabolism is also very interesting for me. We're more focused on mitochondrial function, but energy metabolism during aging is very important. We also use models, and when it comes to brain research, you have to use models because it's difficult to use humans. But you have got these questions many times, but I would like to ask you anyway. So all your nice results on models, do you think they can be transferred directly to humans, or will there be differences?
Well, I would hope so. In fact, this is one of the reasons I really got involved in starting a spin-off from the lab. Because at the end, I'm a medical doctor. I'm very happy to have published many, let's say, nice papers over the years. But, my goal at the end would be to cure people. And I personally think that with the glucose transporter 1 deficiency syndrome, we have a very good model, and we have effects there. And now one of the molecules is ready for what is called IND enabling, meaning that next year phase 1 studies can start. So maybe within 2 years we will know whether we can transpose these models to humans and maybe even cure at least these very, orphan disorders. But I think which will be instructive for other diseases such as, potentially Alzheimer's, but a condition, I don't call it a disease because I'm getting into that category, but aging, it's clear that there is a decrease in glucose utilization with age. And, providing more energy would be, I think, a very, positive, protective approach. Now, it's, for example, if you look at many of the markers, we looked at human brain expression, for example, of LDH, lactate dehydrogenase, and we found essentially the same distribution. In other words, with LDH5 present only in astrocytes and neurons expressing essentially only LDH1. So this gives you just a marker that there is a more glycolytic activity in astrocytes and a more oxidative activity in neurons.
I fully agree with you. I'm also convinced that it will be very helpful for many diseases in the brain. But very often when I'm at dinner parties and people know that I'm aging researchers, they will ask you, do you take supplements? If you do, why? If you don't, why not? And so there's a trend now that these supplements that we're doing research in to treat diseases that maybe will pop up during aging, that we actually start earlier. And I guess it's to prevent them. What is your view on that? Is that a good strategy, or is it waste of money? Or?
No, well, again, as you said, everyone has her or his favorite supplement for the moment.
Or a combination.
Or a combination thereof. But based on actually work from many sides, clearly, again, NAD, which is necessary to produce NADH, and usually NADH is produced by the conversion of lactate to pyruvate. So there is a rationale there for maybe a combination of NAD and lactate. And there is also some evidence for antioxidant approaches like N-acetylcysteine. There is also acetylcarnitine has been, to boost the mitochondrial respiration, but for that you need to provide fuel, so you need to provide lactate. So I think most of them, anyway, at least those that I cited, I think don't have any side effects. So I would take a chance and take some of them. By the way, I take some of them.
Why? Yeah. But that's a good point, and many colleagues are taking different combinations. Often it's supplements that is directed to their own research. But I agree with you that probably most of them are not harmful. However, there are more and more drug companies developing new drugs which are not supplements, they're not dietary supplements, but direct targets to, for instance, these pathways. So can you foresee some challenges in the future with this combination, and how do we communicate this to the population? Because one thing is that we are scientists and we can go and read the publications, we can see if there's evidence or not, but how do we communicate this to non-scientists, because it can appear like a jungle sometimes.
Yeah, so a couple of points in your question. So the first one, the interest from pharma. I agree with you. Obviously, pharma is not interested in supplements because they're not patentable. Now, as you can imagine, with it's, by the way, it's opened up a completely new facet in my activity. I've interacted now with some pharma or even VCs because we have a molecule. It's I can tell you, pharma is still possibly things are changing a bit now, but pharma is still very much in the let's say, mental approach of receptor and interaction, drug receptor interaction, and that's the pharmacology, receptor pharmacology. When you come with ideas of, improving the cellular fitness, the cellular resilience, by, for example, providing more energy, or molecules that contribute to energy, or to other, let's call them quote unquote non-specific, non-receptor specific approaches. And if on top of that you tell them that you're interested in glia, I can tell you is an uphill conversation. Because for pharma it's neurons and receptors on neurons.
Important points. I wonder if we could have some communication from the audience.
My name is Ingrid Amelem, and I work at Oslo University Hospital. And first of all, I would like to thank you for an excellent talk. It was very interesting. And my question is related to lactate as a therapeutic agent, and whether the astrocytes can produce enough lactate from the glycogen storage, or if it's necessary to add extra as a supplement or by doing exercise.
Yeah. That's a good question. By the way, it's an interesting point that you mentioned, can they make enough lactate from glycogen? Actually, as I showed, they can make also from glucose through aerobic glycolysis. But no, your point is well taken, because there is, honestly, as far as I know, not fully clarified whether there is a futile cycle for glucose in astrocyte, like it seems to be in muscle. In other words, that glucose has a cycle through glycogen first before being mobilized. So that's like a buffer, if you wish. So that's still an open, but anyway, it's just a side comment. Is there enough? I mean, there is evidence for a decrease in glucose utilization with aging. So that must be then there is a decrease in lactate production. So now it depends if it's no longer aging but neurodegenerative condition, then of course you lose many of the consumers of lactate because you lose neurons. So you may actually have paradoxically an increase in lactate levels, but I think this is when neurodegeneration is well established and ongoing. And so then you can question, whether really to use it, is there necessary for a supplement? I think so, but anyway, certainly in the early stages, aging and early stages of, let's call it dementia or minimal cognitive impairment, yes, and for sure in this genetic disorder, GLUT1 deficiency syndrome, there is a decrease in glucose and lactate in the brain and in the CSF.
And one point is probably that you have to get the lactate delivered when it's needed and not sticking around all the time.
That's a good point also. We know that the brain works, it's very parsimonious.
We had a question here. Could you please identify yourself?
Yes, my name is Torgeir Landvik. I've been a general practitioner. And I wonder, in neurocystic sarcosis with Taenia solium larvae in the brain, they have an anaerobic metabolism of energy and producing lactate. And I wonder in what way will that affect brain function?
Yeah, there is... in fact, Linda is an expert, I guess, on this. But anyway, what I can answer is that, one of the rationales for trying, I have to say, it was a bit of a trial, lactate in the animal models of depression, was that meta-analysis, epidemiologic analysis shows that exercise is actually an antidepressant in a way. Of course it varies. But overall, on a large number of cases, many studies. It's clear that it pops up as a clear protective factor. And that is one of the elements. I don't think I mentioned it. So there is also, even extreme exercise like X-Fit or something like that. I mean, a very strenuous anaerobic exercise that leads to lactate production. But there are also studies, and particularly from Japanese group, Hideaki Soya and others at Tsukuba University, showing that actually even exercise, mild, moderate exercise that doesn't actually change the lactate index, the oxidative index in the plasma, already has positive cognitive effects. And in a way, it's not surprising because when the brain is active, you don't need necessarily to move, very strenuously muscles. I mean, just do some movements. You will have to activate sensory motor pathways that will release glutamate and activate these ANLS. So exercise, even a moderate one, is probably good.
Of course, exercise is good in many ways. In addition to not only by producing lactate, but also in other ways. And
of course.
They will be together. We have several questions here. We have Imen Belhai.
Hi, I'm Imen Belhai from Bergersen's lab, PhD student. Thank you for a great talk. I was wondering if there are different subtypes of astrocytes that contribute to different roles of this lactate metabolism. And also, I have a second question. If you know if age-related changes in the astrocytic function do contribute to neuroinflammation and diseases like Alzheimer's disease? Thanks.
Yeah, thank you. These are again two very good points. One, regional or cellular specificity. I think there is no doubt that there are different types of astrocytes expressing different properties, but I think some fundamental ones, like glutamate uptake, I guess it's quite conserved. I don't know if there are quantitative and regional studies. I would think that some of the groups here in Oslo did it. So there may be specificities depending on the region or subtype, but maybe some of the fundamental functions are maintained, I would think. Then the second part, the inflammation, is very interesting. I mean, we ran studies with exposing astrocytes to different pro-inflammatory conditions, TNF-alpha, IL-1 beta, for example, and really they change definitely the metabolic phenotype of astrocytes. So inflammation clearly, and essentially what it does, let's say the term that we used was make astrocytes a bit more selfish. In other words, they probably have to cope with many activation of many pathways, and so they are less prone to release, to take up, I mean, the glucose they take up and the glycogen they break down, they use it for themselves more. I mean, I'm simplifying a bit, but that's the general message.
Yes, my name is Christian Pritz. I'm a biochemist at the Department of Biosciences at the University of Oslo. So this was very interesting to hear. I wondered, during long-term starvation, when your glycogen and glucose is all gone, do the astrocytes play a role then? You can live from partly from ketone bodies, and how is the cooperation between the cells then?
Yeah, this is obviously a very important point, and it relates to, for example, the ketogenic diet. Clearly, you can starvation. Okay, it can be just starvation, but starvation often is also, I mean, starvation. It's really glucose free, essentially, carbohydrate free diet. And there are, again, many metabolic adaptations. For sure, the expression of monocarboxylate transporters in the blood-brain barrier increases, so more lipids, free fatty acid, can actually enter the brain and be used by neurons directly. I mentioned the GLUT1 deficiency syndrome. Actually, the only treatment that actually works in the absence seizures of these kids is the ketogenic diet. The only problem, this is a very difficult diet to implement, because essentially you have to eat cream and butter, and that's all. So it's not so easy. So going to your question, yes, there is definitely adaptations, and certainly in neurons. In astrocyte, I would think so. I haven't really studied this, but in the blood-brain barrier, yes, an increase in transporters for, MCT, monocarboxy, MCTs, because they also transport ketone bodies, not only monocarboxylates.
Hi, I'm Johanne Egger-Rindholm from the Department of Molecular Medicine at the University of Oslo. Thank you for a very nice talk. There is some evidence that other glial cells, like oligodendrocytes, also have a high glycolytic activity. So my first question is, do you think that there is a collaboration between astrocytes and oligodendrocytes in their supply to neurons, and how might that collaboration be? And the second question is more related to humans that have a different glial to neuron ratio. How would that affect the supply of lactate?
Yeah. So absolutely. In fact, a few things. First of all, I think the work of Klaus Armin Nave in Göttingen has undoubtedly showed that oligodendrocytes, being also glycolytic, they are necessary to maintain axonal integrity. So in a way, it's a very smart. I have no doubt. I think there is an oligo-axon lactate shuttle. And it can be direct, as Klaus Armin has shown. Could also be indirect, because, as you know, astrocytes are connected to oligodendrocytes through connexins. So it is possibly that, the astrocyte take up glucose, transform it glycolytically to lactate, and then lactate is released at the axon. So anyway, whatever mechanism, probably both or mainly the oligo one based, is a very smart way to provide energy to axons, myelinated axons, and so absolutely. And by the way, a few years ago we published a paper with Jeff Rothstein in amyotrophic lateral sclerosis models and patients, and what we showed is a decrease in MCT1, mostly actually in oligodendrocytes, and so yeah, absolutely, I agree with you. The second one, in terms of ratio, maybe the only comment is that makes even maybe more relevant to your first question, to humans. Because the ratio obviously implies a higher density of astrocytes than in other species, in humans.
One thing we haven't touched directly upon is the role of the receptors for lactate, because since lactate is so important, yes, we would expect there to be a receptor, a sensor, yes, to monitor how much is there around.
I agree. Obviously, this is something very interesting. I know, Linda, you worked on this, on the HCAR and CGP, GPC cap, GPC 81. These lactate receptors. So they are there, of course. They seem to, in some cases, I mean, this work from one of my former postdocs, Jean-Yves Chaton, to actually have an inhibitory. When lactate acts on these receptors, it tends to inhibit firing. I have to say, in all of the essentially data that I presented, we always tested for whether these effects were antagonized by antagonists of this HCAR, these lactate receptors, and we didn't see any effect. So for us, I mean, there probably are two signaling mechanisms. One is through the transporter, and what happens inside the cell, and then others may be mediated by the receptors, but there is less known, I have to say. There is even a receptor in the locus ceruleus, which it's not clear what it does, what type of...
It's a different one.
There is something there, certainly a nice area of investigation, but I think despite the fact that there is evidence over the last decade, not much progress has been done.
One problem with the lactate receptor is that there isn't any good antagonist.
Yeah.
There are agonists, so you have to work with knockouts.
And the pharmacology is not the best.
Not so good,
Andrew Whitelow, a newborn brain injury at the University of Bristol in the UK. I was very interested in your discussion on lactate and long-term potentiation. And I wondered if there was any evidence in humans that lactate administration in any form potentiates learning, whether there have been experiments with university students, for example. Does it help learning in any way?
What I know is actually there have been studies, I think mostly by Danish exercise physiologists, looking at cognitive performance after exercise, and so associated with increased lactate, circulating lactate, and the far as I know, it's not my field, they seem to perform, let's say, better in certain tests. That's, I think. I don't know if administration exogenous, but maybe Linda can comment on this.
I think Linda would be the best expert to talk about this, but certainly I think, like you say, Pierre, that exercise increases cognitive performance. It's also protective against Alzheimer's, and we believe that lactate has a role in this. I think you should specify it more, Linda.
Yeah. Thank you very much. We have gone over the scheduled time.